For most of the past 100 years, the way we've studied cancer metabolism is to take cells from a tumor, grow them in a dish in the lab, and study their metabolism in the dish in the lab. And we've learned a lot by doing that, but we haven't actually been able to answer the fundamental question of which metabolic pathways get activated in an actual tumor that's growing in a patient. Every patient that was recruited to the study, and these were all patients that had lung cancer, we imaged their tumors using several different forms of MRI. And we use these different techniques of MRI because they tell us about the different biological properties of the tumor. Then the patient came in to have the surgery. The tumor and the surrounding lung tissue was going to be resected from the patient. And at that time, the patient was infused intravenously. Basically, we set up a drip with a form of glucose that was labeled with a label that could be tracked through metabolism in the tumor. After the surgery, we could analyze the metabolic activity in each of these individual pieces of the tumor. And we not only analyzed metabolism, but we also looked at which genes had been turned on, which genes had been turned off, and what the tissue looked like in terms of how many cancer cells were present in the tissue and how rapidly they were proliferating. If you look at the way that tumors use glucose, and again, we're working with patients that have lung cancer, and we're analyzing the metabolism of their tumors as that metabolism is happening in the body. Individual tumors are quite different. So some tumors are actually quite aggressive in the way they take up glucose and generate energy from that glucose. Other tumors don't seem to care quite as much about glucose. So that was actually quite eye-opening. Even if you select different areas of the tumor, they're remarkably different. So the way that those individual neighborhoods within the tumor consume glucose and use it to make energy is quite different from region to region. And what we learned by doing our image analysis was that we could actually use the imaging to predict which areas of the tumor were using predominantly glucose versus predominantly another fuel. So that's very exciting because I think that really opens up a whole new world in terms of the way we analyze cancer metabolism.